Welcome everyone, and good afternoon. Thanks for joining us for the third session in the eDNA webinar series. My name is Sam Semino, and I'll be moderating today's webinar. I work for the USGS, where all of my time is dedicated to the Pacific Northwest Aquatic Monitoring Partnership, or PNAMP for short. PNAMP, along with StreamNet, co-led the planning of this webinar series, and the planning group included 13 people from a variety of organizations, including David Pilliod, who organized and contacted all of the speakers for this eDNA webinar series. Today, we will be concluding the eDNA webinar series with the eDNA 301 presentation, multi-species and biodiversity assessments, focusing on laboratory procedures and interpretation of results, including challenges and future directions. This will be presented by Taylor Wilcox. If you missed the previous two eDNA presentations, the recordings are available online on the PNAMP YouTube channel and linked to the Emerging Technology Information Session project page at pnamp.org. Here's today's approximate agenda. After I wrap up the welcome, we'll do an icebreaker question to make sure everyone is awake after lunch and familiar with using some of the functions of Microsoft Teams. We are planning on about a 45-minute presentation from Taylor Wilcox after the icebreaker and opening. And then with the remaining time, we'll do question and answer and discussion. But first, some quick tips on navigating the meeting platform. Please do mute yourself when you're not speaking. If you're on the phone, you can use *6 to mute and unmute yourself. If you're using your computer for audio, you mute and unmute yourself using the microphone icon on the toolbar. Microsoft has been rolling out a few changes to MS Teams lately, so how you get to your toolbar will vary depending on which version of Microsoft Teams you're using. In the older versions, you can make the toolbar appear by hovering your mouse over the meeting window. And in the newer versions, the toolbar is always present. It's fixed at the top right of the meeting window. But regardless of the version, the icons are the same. However, yours might be in a different order than what I have on this slide. So a couple additional tips. If you're having trouble with your audio, check your device's settings, which you can get to from the 3 dots or the ellipses on the toolbar. And you can always use the meeting chat to contact the organizers, which is myself, or my coworker, Amy Poles, and we'll do our best to help you out. When it gets to the question and answer and discussion session, if you have questions, you have 2 paths to ask them. You can type your question into the chat and then I will read it aloud, or you can click on the hand icon on the toolbar and raise your hand and we'll call on folks to unmute their audio to ask their question. So now a little warm-up icebreaker question, and to use some of these, we're going to do a quick interactive poll using Mentimeter. So if you take a second to open up the chat, you'll find that my coworker Amy has posted a link to an icebreaker question. Again, to get to the chat, click on the speech bubble icon to open the chat sidebar, and you should see a link to the icebreaker question that Amy posted. If you would humor me and take 30 seconds to fill out that poll, I'd really appreciate it. And I will switch over to that. We will see some live results coming in. Cool. So the question, how much DNA do you share with a mouse or a fruit fly and a banana? And this is, of course, assuming that you are a human answering this question. We'll give you guys another 10 seconds or so. We're definitely getting quite a few Answers rolling in. And again, if you can't find the chat, you can also go to menti.com and then type in this code at the top, 132355-2, and it'll take you to the same live poll. And I have the answer for this. And it actually looks like our average is pretty close. So I will switch back to our PowerPoint. And there you have it. So we actually share about 85% DNA with mouse, 49% with fruit fly, and 41% with a banana. So we are basically right on with the mouse and really close with the fruit fly and banana. Yeah. Now, while Taylor's sharing his screen, I will introduce him. So if Taylor, if you want to take control. Yeah, Taylor Wilcox is a research geneticist at the National Genomics Center for Wildlife and Fish Conservation, a science collaborative with the USDA Forest Service's Rocky Mountain Research Station, focusing on emerging molecular tools, including conservation genomics, forensic genetics, and environmental DNA sampling. He lives in Missoula, Montana. Go ahead, Taylor.
All right, thanks, Sam. Yeah, so I'm excited to be giving the summit or the webinar today. I just want to outline some communication goals from this webinar so you know what you're getting yourself into. When coming away from this, I would like for people who are listening to be able to identify questions that are a good fit for a multi-taxa analysis approach. We're talking about environmental DNA sampling. To be able to describe how metabarcoding works, including the goals of the bioinformatics steps, just at a very general level. To be able to communicate some of the key considerations and inferential limitations associated with metabarcoding. Again, this emphasis on metabarcoding because it's really the dominant multi-taxa eDNA analysis approach currently used. But then also be able to come away aware of some emerging alternatives to metabarcoding. They're either just starting to be applied or on the horizon. So my goal is to get to these 4 points. Oh, actually, I was going to remind to have the poll here. So I think Sam's going to put the link to the poll there, and then I'm going to go to the view of that by sharing on my side. All right, looks like it's stabilized in the mid. Well, I'm going to call it a good enough sample probably at this point. So it looks like most folks have. There's it's really across the distributions really though. I mean, if you're looking at the averages really. So hopefully there'll be something here that's applicable to folks who really are experts with eDNA and high throughput sequencing and metabarcoding, but also accessible for folks who are new to the field. I'm going to go ahead and swap over to the slide again, and then there's just only going to be one more poll in this talk near the end. All right, so for an outline, first I'm going to introduce a little bit, kind of talk about what questions require a multi-taxa approach. So compare this with a single species approach that Carl talked about last week. Then we're going to spend the bulk of the time talking about metabarcoding, getting down into the nuts and bolts of how it's actually done. We're basically going to walk through a generalized metabarcoding pipeline. So we're going to talk about amplification, some of the key considerations there. We're very briefly going to touch on library preparation, have a little intro to synthesis by sequencing by synthesis, if you're not familiar with that. Then we're going to talk about bioinformatics, again, using kind of a generalized pipeline that we'll walk through that I think should be applicable to a lot of different individual packages and tools you might use. Talk a little bit about interpretation of metabarcoding data, and then try and transition from there to a kind of a broader discussion of kind of interpretive challenges and limitations that are general to multi-taxa eDNA sampling approaches. I'm finally going to spend a couple of slides just bringing up some alternative emerging technologies, not getting into the details of how those work, but providing some links or some citations so you can follow up on those, and then hopefully leave a fair bit of time at the end for questions. Well, I was excited to agree to talk about this part because the eDNA 3.301 because this is one of the more interesting, kind of exciting things about environmental DNA, right? The idea that you could have a cup of water and in there is genetic material from megafauna, from fish, from nematodes, from bacteria, from viruses. It's just really a neat idea. And I think that really environmental DNA sampling is unique in the level of taxonomic scope that you potentially have with a sampling event. The fact that you can cover so many broad taxonomic groups with your sample. At the same time, you don't always need an analysis approach that is going to be that broad as your sample is from the environment. You may be better suited to a single species approach if you have a highly targeted question where you have a low tolerance for errors, both false positives and false negatives, and also where you have a limited budget and you really need to constrain the analyses that you do to the specific questions of interest. But there are some cases where your question is inherently multi-taxa or inherently calls for a multi-taxa approach. I think there are three main categories that this falls into that I can think of, but maybe other folks can think of other examples during the discussion time. One is if you have a potential novel species that you're interested in detecting. So for example, let's say we're looking for invasive species. If your question is, are there northern snakehead in this river system? Then you have a question that is targeted and you can use a single species test to answer that. You can have a northern snakehead qPCR assay, for example, and see whether or not their DNA is present. But if your question is more broad, it's something like, are there invasive fishes in this system? You have this kind of undefined species list. You're interested in many different species. You couldn't put your finger on one specific species that you're looking for. And in that case, you really need a multi-taxa approach that is going to look across fishes rather than look for individual species that you have on your focal list. Another case is where your species list is very large. So you may be able to identify each individual species that you're interested in, but there's just too many species for it to be practical to design single species tests or to implement those for your samples. So an example of this might be an index of biotic integrity. Here you have a list of taxa that you would be able to put into your index of biotic integrity, so you know what your species list is, but that's a very large species list, and it's probably not practical to design individual species-specific analyses or to conduct those on samples. And then finally, I have a category that I'm just calling taxon-specific assays and practical. So this is kind of a catch-all for special cases where maybe you're able to identify individual species, but designing of species-specific assays is not going to work because there's phylogenetic uncertainty, or it's a really species-diverse system with lots of really closely related species, and so you're not able to design species-specific assays. But there are these cases where it's simply impractical or impossible to apply a taxon-specific analysis, which again pushes you into a multi-taxa approach. All right, so that's the first part of the talk, just introducing this idea of why would we go to an environmental DNA 301 in the first place? What are some of the questions that are going to call for this analysis approach? Next, we're going to talk about metabarcoding. And as I talked about in the very beginning, metabarcoding is the predominant multi-taxa analysis approach for environmental DNA sampling right now. First, I'm going to introduce a little bit of what metabarcoding is, so we're going to understand why I'm talking about the individual steps following there here in the outline. So metabarcoding really builds on this genetic approach called barcoding. Barcoding is where you have a barcode region, so you have a locus, a portion of the genome, typically from the mitochondrial, which you're amplifying using PCR, which Carl talked about. So you're using primers, multiple cycles of heating and cooling to make many copies of a specific region of the genome. And then you're sequencing it. And then based off of that sequence, you're able to figure out what species that genetic material actually came from. So that's barcoding. Metabarcoding simply recognizes that you have samples, like an environmental sample, where you have genomes not just from one species, but where you have genomes from potentially many different species. And you can use the same primers, these might be called universal primers or conserved primers, in order to amplify this locus, in order to amplify this genome region across all of, or many of those different taxa. So in this case, we're representing 4 different mitochondrial genomes with 4 different colors. It's the same locus, the same mitochondrial genome region that's being amplified by the same primers, but there are sequence differences between these. And based off of these differences in sequence, we're able to infer the different species from which this genetic material came. So that's basically the idea behind metabarcoding. You have bulk amplification with conserved primers of a barcoding region across multiple taxa. You use high throughput sequencing, which I'll talk about a little bit more in a minute, in order to sequence those amplicons. Based off of those sequence identities, you compare those sequences with a reference database where you know the sequences for a list of species, and from that you're able to infer your species composition. All right, so we're going to walk through now the individual steps of how you do that in a little bit more detail. First, we're going to talk about the amplification process, so the PCR. Specifically, I want to focus on primer selection. I'm going to focus on 3 different attributes here of primer selection. First is coverage, that is having efficient amplification across all of your target taxa. Excuse me. The second is off-target amplification. We want primers that minimally amplify non-target DNA, and I'll talk about why in just a little bit. And then finally, we want a high level of taxonomic informativeness. That is, that the actual sequences that we get from this effort are going to allow us to identify species without ambiguity. So I'm just going to walk through each of these one by one. I'm going to use cartoons in order to illustrate what I mean by these. Hopefully this will be helpful for folks who are a little bit more visual. So I'm first going to introduce that cartoon using a basic metabarcoding enrichment example. So here we have a population of templates, so of genetic material, and we have 2 different templates that I put here. The blue are the non-target and the orange are the target templates. And then what we're going to do is we're going to imagine having some cycles of PCR. So we're going to amplify our target DNA, and we're going to track the abundance of these different templates as we proceed with PCR. So here, bottom left, I've put a plot. So we have PCR cycles on the x-axis, the on the y-axis. We have an orange dot in the bottom left indicating our target template, blue in the top left indicating our non-target template. And now we're going to do some PCR. So we have these red oligonucleotides, these short line segments that are annealing to our templates. And we're going to do a cycle of PCR. So there's no increase in the abundance of our blue non-target template, but there's a doubling of our target template, another cycle of PCR, and another cycle of PCR. And what you can see we've done is by applying PCR, we've massively enriched specifically for our target template, so that now after PCR, our template pool, our PCR products are dominated by our target. So when we go to sequencing, what we're largely going to get are target sequences. We're going to get those portions of the genome from the specific taxa that we are interested in for our analysis. So now I'm going to use the same cartoon to talk about the idea of coverage, the idea that we need to have efficient amplification across all of our target taxa. So to understand this a little bit better, let's use the same kind of way of looking at how PCR works, where we have our primers that are annealing to a template DNA. And when we're using conserved primers, we're taking advantage of the fact that not only are there similar sequences among different templates that we're going to amplify with these primers, but these primers are actually able to anneal and amplify the DNA even if there are base pair mismatches. That is, even if the primers are not exact complements to the template DNA. Now, the thermodynamics of this are a little bit complex, but at a simplistic level, you can imagine that when there is a perfect match between the primers and their template, there is a high level of amplification efficiency. As you increase the number of base pair mismatches, that amplification efficiency decreases. So if you have high amplification efficiency, you have a close to doubling of your PCR products with every cycle. If you have a low amplification efficiency, then you have a less than doubling, potentially a very slow increase in the abundance of your templates or of your products over time. So let's go back to this cartoon with that kind of idea in mind, that we have potential variation in our amplification efficiency. We have our non-target templates again here in blue. We have our target 1 in orange, and now we've introduced a new target indicated in green. Again, we're going to use this plot with PCR cycles on the x-axis and amplicons on the y-axis to track the abundance of these products over time. We have green on the bottom, orange just above it, and then blue at the very top. We'll ignore blue as we move forward. But in this case, now what we're going to do is we're going to imagine that for target 1, our PCR isn't as efficient as it is for target 2 in green. So there's not going to be a perfect doubling of the orange, but there is going to be a perfect doubling of the green target. So after one cycle of PCR, you can see there's an increase in the abundance of both templates. Another cycle of PCR, you can see that that template 2 is starting to catch up. And then especially as we start getting into later cycles of PCR, you see this shift where there's this over-enrichment of target 2 relative to target 1. So the problem here is that if this proceeds far enough, we could have target 2 dominating our post-PCR pool of PCR products. And when we go to sequencing, we might actually completely miss target 1 and only pick up target 2. So because there's a difference in the amplification efficiency for these two targets, we have reduced sensitivity for target 1, and we might miss its detection as target 2 is over-amplified. All right, so now we're going to talk about kind of the inverse of this, which is off-target amplification. We use the same cartoon. We're going back to this simple case where we just have one target template and we have one non-target template, which is indicated in blue. We're going to track them over time, but now we're going to imagine that these universal primers are a little too universal and they're actually binding to DNA that we're not interested in and causing some low level of amplification there as well. So if we look at the abundance of these amplicons over time, we see that as we increase our number of PCR cycles, increase in in the PCR products, but we not only have an increase in the PCR products for our target DNA, but also for our non-target DNA. And this is a problem for the same idea as we had before, that we could still have our non-target DNA dominating our sequencing pool. We could end up sequencing a whole bunch of DNA that we're actually not interested in and missing those specific taxa that we're hoping to amplify, that we're hoping to enrich for with PCR. All right, and then finally we're going to talk about consider taxonomic informativeness. Here I'm going to have to use a different cartoon to articulate the idea. I want to introduce this idea of a barcoding gap. So I'll talk about it here, but then I'm going to break it down with the cartoon a little bit further. What we need to do is we need for the intervening region that's amplified by PCR to result in a sequence where our intraspecific variation, that is the genetic variation within one taxon, is less than the interspecific variation, that is the variation between individuals of different taxa that are being amplified. And if there is this gap, there's this difference, then we're able to unambiguously assign sequences back to taxa. But if there's an overlap between our intraspecific and our interspecific variation, then it's not possible for us to always unambiguously assign sequences to taxa. Let's go to our illustration of this. Here, we're imagining that there are 4 different taxa, taxon A, B, C, and D. We have 4 individuals of taxon A, 4 individuals of B, and 1 each of C and D. We're looking at a portion of a imaginary amplicon sequence from a barcoding region. We have a consensus sequence on the top, and then any place where those base pairs differ for individuals are indicated with an underscore below. And let's get a feel for what we mean by intraspecific and interspecific variation. So let's look at taxon A only. So we're only going to be considering interspecific and intraspecific relative to taxon A. Our level of intraspecific variation, that is the number of base pair mismatches between any 2 individuals of taxon A, ranges from 0 to 1, versus our interspecific variation, that is any individual from taxon A to any individual of taxon B, C, or D, ranges from 4 to 7. So if we were to plot a histogram that looked kind of like that introductory figure to this topic, we have genetic distance on the x-axis and frequency on the y-axis. We would see that there is indeed a barcoding gap. That is, our intraspecific variation is in a distribution that's lower in genetic than our interspecific variation. So we can unambiguously assign any sequence from taxon A back to that taxa. All right, now let's look at an example where we have ambiguity. So here we have a case where there are some individuals of taxon A where the sequence is more similar to individuals of taxon B than to other individuals of taxon A. So there's this this problem where we have actually greater levels of intraspecific variation, that is variation within taxon A, than variation between some individuals with taxon A and taxon B. So in this case, some of the sequences that actually come from taxon A are going to be ambiguous. We're not sure if they really should assign to taxon A or to taxon B. And obviously this is a problem if the whole goal is to infer what taxa you actually have present. All right, so we talked about the idea of having high levels of amplification efficiency across all of your templates, about minimizing non-target amplification, and about having an amplified region that's taxonomically informative. The big takeaway that I want to communicate from this is that primer selection is really critically important. There's probably no one primer set that's going to be the best. The one, for example, fish metabarcoding primer set that you should use for all questions. You are probably going to need to match the primers that you use to your specific question. So we can talk about this more during the discussion period, but for example, primers that have high taxonomic coverage tend to have lower taxonomic resolution. So there are trade-offs when you're selecting your primers. The good news is that there are some useful tools out there for assessing primers and for designing primers. I've laid out this as kind of a general way in which you can think about validating or developing primers before you go to your metabarcoding project. You can either take primers that are already published in the literature, or you might want to develop primers specifically for your question. In either case, you're going to use some similar software for assessing these relative to a reference database. This is a non-exclusive list, but some common tools are EcoPCR, which is a Python-based program, Decipher, which is an R package, and Primer3, which is a standalone system. You're going to use this software to assess your oligonucleotides that came from the literature, or to develop new potential primers relative to a reference database, which again, you're going to need to develop specific to your system. So maybe you're sourcing these sequences from something like NCBI GenBank, but you're going to be specifically selecting sequences that are relevant to your system. And then once you've done this kind of in silico assessment of the primers against this reference database, you'll want to test those primers against known samples. So this is probably tissue extracted DNA initially, and then environmental samples from well-described communities to see how well your primers are actually performing. All right. The next step, if we're doing metabarcoding after amplification, is library preparation. I'm going to walk through this one super fast and not talk about nuts and bolts here very much. But for those who are not familiar with genomics generally, library preparation is essentially the process by which you take samples and prepare them for high throughput sequencing. One of the key steps here is that your samples need to be molecularly tagged, and you'll see this in the sequencing by synthesis video here in a minute, in such a way that your samples can be pooled onto a single sequencing lane. So I am just going to make one note about this molecular tagging specific to environmental DNA. If you do genomics generally, there may be ways in which you are used to preparing your libraries, which result in low levels of transition of sequences from one index library to another. So you have misassignment of your sequencing clusters to the wrong library, and which may not matter that much for a lot of population genomics analysis where you're discarding really low abundance reads. But in environmental DNA sampling, you're often interested in things that are really low abundance. And so you may need to think carefully about your library preparation in order to minimize this false positive or contamination risk during library prep and sequencing. So that's the takeaway I want to get from library preparation. Not going into the details, but it is important to think about potential crosstalk between samples on the same sequencing lane, and to take steps to mitigate that in your library preparation and potentially in your bioinformatics. All right, next I'm going to talk about sequencing. So for folks that were near 10 on metabarcoding and high throughput sequencing, you can tune out for a little bit, but I think it's helpful to understand just how Illumina sequencing platform works. And I'm talking specifically about Illumina sequencing because that's our predominant high throughput sequencing approach used right now. So for that, I'm going to attempt to use a video from YouTube that's actually put out by Illumina that I think does a pretty good job of illustrating this. This will take a little under 3 minutes.
DNA fragments. Through reduced cycle amplification, additional motifs are introduced, such as the sequencing binding site, indices, and regions complementary to the flow cell oligos.
And I'm just going to pause there for a second because I skipped ahead just a little bit, and note that when we're seeing these two strands, that's the same kind of representation as I was showing with the two parallel lines in the PowerPoint. So these are template DNA that's then been amplified. So these are PCR products now, and they're talking about actually appending these small sections of DNA to the ends, which have an index sequence and an adapter sequence.
Regions complementary to the flow cell oligos. Clustering is a process where each fragment molecule is isothermally amplified. The flow cell is a glass slide with lanes. Each lane is a channel coated with alon, composed of two types of oligos. Hybridization is enabled by the first of the two types of oligos on the surface. This oligo is complementary to the adapter region on one of the fragment strands. A polymerase creates a complement of the hybridized fragment. The double-stranded molecule is denatured, and the original template is washed away. The strands are clonally amplified through bridge amplification. In this process,
oh, sorry guys.
The strand folds over, and the adapter region hybridizes to the second type of oligo on the flow cell. Polymerases generate the complementary strand, forming a double-stranded bridge. This bridge is denatured, resulting in two single-stranded copies of the molecule that are tethered to the flow cell. The process is then repeated over and over and occurs simultaneously for millions of clusters, resulting in clonal amplification of all the fragments. After bridge amplification, the reverse strands are cleaved and washed off, leaving only the forward strands. The 3' ends are blocked to prevent unwanted priming. Sequencing begins with the extension of the first sequencing primer to produce the first read. With each cycle, fluorescently tagged nucleotides compete for addition to the growing chain. Only one is incorporated based on the sequence of the template. After the addition of each nucleotide, the clusters are excited by a light source, and a characteristic fluorescent signal is emitted. This proprietary process is called sequencing by synthesis. The number of cycles determines the length of the read. The emission wavelength, along with the signal intensity, determines the base call. For a given cluster, all identical strands are read simultaneously. Hundreds of millions of clusters are sequenced in a massively parallel process. This image represents a small fraction of the flow cell. After the completion of the first read, the read product is washed away. In this step, the index 1 read primer is introduced and hybridized to the template. The read is generated similar to the first read. After completion of the index read, the read product is washed off and the 3' ends of the template are deprotected. The template now folds over and binds the second oligo on the flow cell. Index 2 is read in the same manner as index 1. Polymerases extend the second flow cell oligo, forming a double-stranded bridge. This double-stranded DNA is then linearized and the 3' ends are blocked. The original forward strand is cleaved off and washed away, leaving only the reverse strand. Read 2 begins with the introduction of the Read 2 sequencing primer. As with Read 1, the sequencing steps are repeated until the desired read length is achieved. The Read 2 product is then washed away.
All right. Let's see if I can progress the slides. So I think what it's useful to understand how the sequencing occurs, because it helps us to understand what actually comes to you from the sequencing facility. So the fact that you're getting separate information for a P7 index or a forward index and a P5 index or a reverse index, and you're getting a forward read and a reverse read, which are potentially overlapping on that single PCR product that you've actually put in. And helps us to understand some of the bioinformatics steps moving forward. All right, so with that, we're going to now talk about, after we've done our amplification, we prepared our libraries, they've been sequenced. Here we're imagining on an Illumina platform. We're going to talk about what we actually do on the computer to try and filter through what we've gotten and come up with a species list. So this is a really great time to be doing metabarcoding because there are, even if you're not a bioinformatician, because there are just a lot of really user-friendly tools out there for doing this. Some of these have been around for a long time or developed for the microbial world, such as QIIME2 and Mother. There are new tools that have GUI interfaces. Some of Galaxy projects have been developed, or SLIM is really specialized in metabarcoding. But I'm going to try and talk about bioinformatic filtering and processing in a way that's general across the many different ways, the many different tools that you might use for doing this. Although there are going to be differences and slightly different orders in some of the steps that you might be able to take. So first, I'm going to give you a look in actual FASTQ files, so what the actual sequence data looks like when it gets sent to you from a sequencing facility. And then I'm going to walk through that generalized bioinformatics workflow, which again, I've outlined here with bullet points. All right, so here we have a directory that has some zipped folders in here with FASTQ files. I've highlighted in red the forward and reverse reads for some paired end reading of a sample called KP-S1. And this is actually not environmental DNA, but this is Fisher scat, but it's not really important for this demonstration. So we have the name, and then we have R1 for the forward read and R2 for the reverse read. You can unzip this and you could just look at this in a text editor, and this is what you would see. There's going to be hundreds of thousands of lines that look like this. Again, I'm highlighting in red now just one read. So if we break down these 4 lines, you can see that the first part of this first line is the run information and the location of the cluster on that flow cell. So you can think back to that video we just saw. The next bit here are the index sequences, our forward and reverse indices. That actually allowed us to identify which library this cluster was associated with. The next is our actual reads, our ATGC sequence that we've inferred. There's a plus on the next line, and then below that is our quality score, which gives us an estimate of how likely it is that that base was called correctly. And this is based off of an algorithm that brings in information about the cluster fluorescence intensity and how close it is to other clusters. All right, so now we have an idea of what that data actually looks like that we're getting. All of our bioinformatics subsequent to that are going to be based off of just those repeated 4-line format that I showed you in the FASTQ file. All right, so I'm not going to break down this first step, but I'm going to recognize that really the first step for bioinformatics starts before where we're going to get into the nitty-gritty, and that is at where you assign your sequences to your library. So I showed you a FASTQ before, which means that this has already been done, and that's typically how you're going to get these data from a sequencing facility. They're already going to use those index sequences based off of a list that you gave them in order to infer which library each of those sequencing clusters came from. But there is some level of kind of bioinformatic interpretation here. You may not require, for example, exact match between the index sequence and the that you provided and that which was actually read off in order to assign a sequence to a specific library. You might filter those indices by quality score. So I'm going to skip over this step because typically it's sufficient to simply receive those library assignments as they come from the sequencing facility, but it is possible to go back to the raw base call information and do this assignment of sequences to library in a different way than is the defaults for the instrument. Okay, next step is to trim off our primers and our adapters. So this here is an illustration that should remind you of the video we just saw. We have some PCR products here. We have in the light blue is the actual sequence that we're interested in. On either side of that are our amplification primers. Then we have a paired adapter that attaches our amplification primers to an index sequence. Here there's an index on both sides, a P7 and a P5. And then finally there's a P5 and P7 tail, which are used for actually attaching this to the flow cell for sequencing. So everything that are in those red ovals on either side are sequences that we've actually introduced into the system. And so we want to trim those off because what we're really interested in is this real biological sequence intervening our 2 amplification primers. All right, once we've trimmed those off, we're going to filter and trim the remaining sequences for quality. So I talked before about how on the FASTQ we have a quality score for each base pair. This ranges from 0 to 40, and that to an error quality or an error probability. So a quality score of 40 means that you have a low probability that that base was called with error, so you have high confidence in that call. A low quality score, like around 10, means that there's actually a pretty high probability of there being an error. You're less confident of that base call. So for example, let's look at the forward read from that KPS1 library that I showed you before. The axis legends here are a little bit small, so I'm going to describe the one on the left here for you. On the x-axis, we have cycle number, so this is base pairs from left to right. On the y-axis, we have quality score ranging from 0 at the bottom to 40 at the top. And then I'm not going to walk through all of the different colors here, but basically we have a summary of the distribution of quality scores at each base pair across all of the sequences in this library. You can see that we tend to have pretty high quality scores for these sequences for the first about 100 base pairs, but then that quality drops off really rapidly. So one thing we might do is trim off all sequence information after a certain number of cycles. And then we might also impose some types of rules where we remove sequences that have, for example, a certain number of base pairs that were called with a quality score of less than 20. So after we've done something like that, we get the distribution that we see on the right, where we have pretty high quality scores across our reads, across our library for the entire extent that we're going to work with. And we filtered out some of those that have really low quality scores early on in those cycles. I'm going to note here that there's a trade-off. If you're too stringent with doing your filtering here, that is, you require that there only be bases that are called with a high level, with really high quality scores, then you end up throwing out a lot of sequence data that's potentially useful, and you're going to reduce your ability to detect rare templates. Conversely, if you're too lax, you could be inferring haplotypes that don't exist. That is, you're more likely to include erroneous sequence data, make inferences that are incorrect. There's no strict rules, but I think it's important to be aware that these trade-offs exist. All right, the next steps might be skipped over in some pipelines, or depending on what you're trying to do, or they might be flipped in order, but you typically are going to merge duplicates. So for example, if you have a read that's ATGC, then you have another read that's ATCG, and it goes on like that. You can do some level of data compression by summarizing and say, I have this sequence ATCG 50 times in this library, rather than reporting them each independently. And then you are going to typically merge your forward and your reverse reads. So remember from that video we saw before, we have a forward read and reverse read. Hopefully these are overlapping on our amplicon. So here, that's what I'm trying to illustrate. Forward read on the left, reverse read on the right, the bold area being where they overlap. Based off of this overlap, we're able to stitch these two together and infer what the full amplicon sequence would be. All right, next, after we've done some of these trimming and quality filtering steps, and we've done some level of data compression, we need to further compress our sequence variance. So if we were to look at a bunch of PCR, a bunch of reads from these PCR products, we would see that for sequences that are really all from one taxon, there's going to be some level of haplotype of diversity here in the sequences. So these differences in base pairs, which here you can see with some of the colors, are a function of both PCR-induced errors and sequencing errors, as well as actual biological diversity, so real haplotype diversity. And what we want to do is find some way of taking these sequences, which are all similar but have some differences, and summarizing them with a single representative sequence. There are two main approaches for doing this. I'm going to introduce them here and then walk through them one by one. The first and kind of traditional way of doing this is called an operational taxonomic unit, or OTU type approach. Here we're going to cluster sequences together based on some threshold distance. So for example, all sequences within 3% of each other are clustered under one centroid operational taxonomic unit. This can be done either closed reference, so where we're taking our sequences and we're assessing them against a library that we've already chosen of what we're going to call our OTU centroids, and we're going to use for representing our operational taxonomic units, or it can be done de novo, specific to your sequencing run, where you're just clustering those sequences around each other. The other approach is called an amplicon sequencing approach, or an exact sequencing variant approach. So this is abbreviated ASV or ESV. And here we're actually going to use our base call quality score information to try and build a model that allows us to infer the original template sequence. So try and remove those PCR and sequencing errors rather than compress them within a cluster. All right, so here's a visual representation of an operational taxonomic unit approach. Here we have 3 OTUs. Let's ignore this yellow chimeric sequence that's been shown here. And these OTUs are representing genetic distance in space. So you have these dotted envelopes around the yellow centroids. And so we're going to cluster any sequence that falls within those centroids as being part of that OTU, and we're going to represent that entire OTU with the sequence that's indicated with the pink dot. One of the things that's illustrated here is that you could have reads that are ambiguous about which OTU they should assign to. All right, then the ASV or the ESV approach actually tries to look at your sequencing data and build a model that estimates the probability that you have errors in your sequence, and then tries to use that information and the distribution of read abundances of different types to actually infer what your original sequence was, so before you introduce PCR and sequencing errors. And these ASV and the OTU type approaches are trying to get at similar things, but I think there can be an argument for why an ASV approach might, in some cases, be better. I guess I hesitate to say it that clearly. There are some downsides as well we can talk about in the discussion period. But here's an example of where you might see some differences that could be important to your inference. So let's imagine here that we have a single OTU. Again, we're representing that with a dotted envelope. We have the OTU centroid here with an arrow pointing to it. And then we have a whole bunch of different reads here that all fall within this single envelope. But what we might actually have are multiple real sequences that are different, that are all being represented by this single OTU. So what we're actually doing here is we're representing some of our reads that are introduced by PCR and sequencing errors, but also some real sequences by an OTU centroid that actually is not a real biological sequence. Versus an ASV approach would try and say, well, let's try and actually estimate what these real sequences are rather than compress into an OTU. And so perhaps an ASV approach would help you to get at cases where you have really closely related species and you need to be a little bit more fine scale with your error correction. All right, so this is, again, the next step is one, again, that could have been done a little bit earlier, potentially, depending on your pipeline. But here we're going to remove our chimeric sequences. So here's an illustration of what a chimeric sequence is. Let's imagine we have some biological sequence A. We have extension in our PCRs, what's occurring here. So the polymerase is coming along, it's synthesizing the complementary strand of biological sequence A, but then for some reason, it's not able to complete that process, and there's abortion of that extension. Then that incompletely extended single-stranded piece of DNA can kind of act like a primer, and it can bind to another biological sequence here that's actually a little bit different. So this is biological sequence B. That's the next part down. And then it can continue with extension. So what you end up with is a PCR product that's actually a hybrid between two different biological sequences, biological sequence A and biological sequence B. So this isn't a real thing in nature, but it's something that's real that we put into our sequencing because this was introduced during our library preparation or during our original amplification. So I'm not going to talk about too many of the details here for detecting chimeric sequences, but most tools leverage the fact that these tend to occur as lower abundance composites of more abundant parent sequences, which I think kind of makes sense if you think about the example above. Again, a non-exclusive list, but some commonly used tools include Slayer, UChime, and there's a tool built within the Decipher package for detecting and removing chimeric sequences. Excuse me. All right, and then our final step in our bioinformatics workflow is taxonomic assignment. And I'm just going to talk about that a little bit at a general level. The idea is pretty simple, where you're going to take the sequences that you have after you've trimmed off your primers and your adapters, you've tried to remove really crappy sequences, you've done some level of data compression, and you've using either an ASV or an OTU approach, and you've tried to remove your chimeric sequences. Now you're going to say, well, what is the most similar sequence in my database of sequences associated with species to this sequence? And that must be the species that this sequence came from. But a real challenge for this is if you have an incomplete database. An incomplete database, then you can fail to assign that read that you have to a species, or you may end up assigning that read or that sequence to a closely related taxa, not the one that it actually came from. So for example, maybe you have a read that is from cutthroat trout, but you don't have cutthroat trout in your database. You might say, well, actually, there's nothing that I can find in my database that matches this sequence. I don't know what it is. So that's a way in which your database incompleteness can cause a problem. You fail to detect cutthroat trout simply because it's not in your database. Or you might find that pretty close to that sequence is rainbow trout in your database. And so you might say, oh, well, this is rainbow trout. This is rainbow trout. When in fact, you're inferring that's a rainbow trout read simply because you don't have cutthroat trout in your reference database. Also note that your sequences may be ambiguous. So you may have, we talked about this some with our primer selection. You could have sequences where you cannot clearly distinguish which taxa they should assign to, or they may assign with uncertainty, where especially in a database where you have maybe not very many representatives, many individuals from each individual taxon. One of the most common bioinformatic tools for doing this, because you have a lot of data to sift through typically, is BLAST. So it stands for the Basic Local Alignment Search Tool, which folks who have a genetics background are probably familiar with. I was going to note that BLAST is a heuristic approach for alignment based on local alignment of smaller seed words within the query sequence. So you don't have to take apart what that means in too much detail, but because of its design, it's pretty reliable and it's really quite fast, and it's been built right into the NCBI infrastructure. And I think this speed and general reliability and the fact that it's built right into things like GenBank and pretty easy to use is why it's been taken up for taxonomic assignments so broadly. But I want to point out that although it's really common, it's not actually designed for taxonomic assignment. It can help you assess how similar your reads are to sequences in the database, but it leaves a lot of room for you to have to come up with interpretation of how close that sequence actually needs to be in order to make a taxonomic assignment. There are other algorithms out there that are built specifically for the purpose of taxonomic assignment. One of the most common is the ribosomal database project, the RDP classifier. This is a naive Bayesian classifier that actually estimates the confidence of your assignment to taxon or to different levels of taxonomy based on a training data set. So where you imagine if you were to get a sequence from your training data set, what your level of confidence would be in assigning it back to taxa. There are some nice kind of innovations on top of this. One, just as an example, is eDNA. This is similar, but it's a machine learning approach that attempts to allow for missingness in your training set and your reference database, which I think is an important consideration and something that you might misestimate your level of certainty if you don't consider the potential for missingness, which is pretty common in databases. But there are many other tools, including in the suite associated with QIIME, MapSeq, Syntax, SpinGo. Centrifuge uses a little bit of a different concept, but has that same goal of getting us back to taxa based off of the sequences that we got. All right, so generally from the metabarcoding portion, takeaway that I want you to get is that there are many reasonable pipelines and thresholds within those individual pipelines for filtering your data, which can be a little bit intimidating at times because it's not, there's not a clear rule. This is the way that you have to do it. I think that one way in which you can increase your confidence is to consider assessing consistency across different pipelines and across different bioinformatic filtering thresholds and see how consistent your results are. I do think that something that you'll see is that taxonomic assignment is one of the most important bioinformatics steps, and particularly your database completeness, because this is really going to decide whether or not you're able to come up with a sequence based off of, or a species based off of the sequence that you've derived, or if you're going to come up with the right species. So considering your database is really important, like I mentioned early on, in setting up the primers that you're going to use, and then in making your final inference. All right, and I see that I'm running a little bit long on this, but I'm going to talk a little bit about interpretation after you've done taxonomic assignment with metabarcoding. And that I think will kind of transition into a talk about interpretation for multi-taxa approaches broadly. All right, so after you've done with your metabarcoding, you've amplified your, maybe this is the best slide to look at, you've done amplification, you've prepared your libraries for sequencing, you sent this to a sequencing facility, you took those sequences, you did some bioinformatic processing to come up with a candidate species list. But there's still some interpretation that's often involved here. And this extra interpretation step often comes because it could become because of taxonomic assignment errors or database issues, or it might simply be the fact that we tend to get low abundance sequences or infrequently sequenced sequences that are unreliable when we're using these metabarcoding approaches. And so it's typical to go through your species inferences that you've made and potentially remove those inferences that are suspect for various reasons. It could be that the reads for these sequences are very low abundance, which suggests that they are contamination or crosstalk between your different sequencing libraries. That you could be removing sequences or reads that lack reproducibility across replicates. So this again suggests something unusual happened, maybe a contamination event with one of your libraries. Certainly removing sequences that are found in your negative controls. Again, evidence of potential contamination or issues in the lab, or simply sequences that are just really highly unexpected. It just doesn't make sense that you're going to have platypus in this part of the country. And so if you come up with that sequence, you may be able to use outside ecological information to identify that as a highly suspect result. I should have included the citation for this supplement to a paper here, but this is just an example of how this can be a little bit of a complex process to think about how you're going to filter and how you're going to interpret after you've done taxonomic assignment. Here, the authors are removing low abundance sequences consistent with how frequently they saw sequences showing up in their negative controls. They're also specifically removing taxa that didn't really make sense, but are to be in their system, but are likely to be contaminants. So for example, some tunas, where that could be something that is a food even that's in the laboratory environment, but it really didn't make sense to have it in their system. And some other pieces of information to try and tease this apart. And again, I think this can be something that's not super satisfying, or can feel kind of hand-wavy as you're going through this. So it's important, in order to feel more confident of your results, to lay out strict rules, to have a tree like this, for example, to understand why you're removing sequences that otherwise seem to infer to species. And I was just going to put this paper up as an example of the fact that depending on how strict you are with your removal of suspect species rules, or what this kind of interpretation process looks like, can really impact your results. So here we have on the left where we look at on the x-axis multiple samples that are collected, and on the y-axis the number of fish species that are inferred from this environmental DNA sampling effort. And we have 3 different levels of essentially stringency in doing this interpretation step. So on the bottom we have a high level of stringency. We're removing sequences that are even relatively reproducible and abundant. On the top we're including we have low stringency. We're including a lot of sequences, even if they're not real abundant or highly reproducible. And in the process, we're coming up with a really different mean total number of species detected. So this is something to think carefully through. Think about what your relative tolerance is for false positive and false negative results. All right, so more broadly, if we think about interpretation with multi-taxa approaches, even beyond metabarcoding, multi-taxa approaches tend to have a more complex detection process than single species approaches, and so this is going to leave more room for error, both false negative and false positive results. So failure to detect species that are actually present and false positive results. And I think if you look in the literature, you'll see this pretty consistently, that to date, multi-taxa tools tend not to be as sensitive as optimized single species approaches. There are always exceptions, of course, but this is a general trend that you'll see and might be one of the reasons that you would think about whether you really need to use a multi-species approach or if a single species approach will allow you to answer your question. And then also we see in these sequencing approaches that false positive sequences, as I described in metabarcoding, are pretty normative. They may be at low abundance, but it may be difficult or completely impossible at this time to entirely remove these low abundance false positive sequences. And you have to deal with them with some sort of bioinformatic or interpretive rules. All right, so I've introduced multi-taxa environmental DNA analysis and where you might use it. We've tried to spend most of our time on metabarcoding, getting into how you actually do that step by step. I warned you that there are some extra interpretive challenges and limitations associated with multi-taxa tools. And then finally, I'm going to rapidly move through some slides on some alternative and emerging technologies to be aware of that might replace metabarcoding at some point or might be complementary. I'm just going to jump right to these slides. One that's really just a building on metabarcoding goes by a few different names. Sometimes it's called multifaceted or microfluidic, depending on the platform, or multi-locus metabarcoding. Here are 3 example publications of that. Basically, the idea here is the same as metabarcoding, but you have multiple loci, multiple genomic regions that you're targeting, or maybe you're using different primers for different taxonomic groups, but then you're pooling all of these together for sequencing. So you're potentially able to get it more broad taxonomically than you could with a single locus, or able to have greater confidence because you have multiple loci per taxonomic group that you're looking at. Quantitative metabarcoding, just very generally recognizing that there are many metabarcoding protocols that folks are working on to try and get estimating organism abundance, not simply presence or absence. Some of the approaches here include known concentration spike-ins, molecular identification tagging of individual template molecules to try and get closer to what your original template concentrations were, models of the amplification process, or template concentration by biomass correctors, which are specific to system. Here again, I'm giving a paper that you might go to as a starting point for reading more about quantitative metabarcoding approaches. High throughput qPCR approach is something we've been working on at the National Genomics Center recently. Here you're using a taxon-specific approach, but you're batching several to dozens of analyses together. So you're doing a targeted approach in parallel. So it's kind of this hybrid between a single species approach and a multi-taxa approach. CRISPR has been in the news because of the recent Nobel Prize being awarded for this work, and we've heard a lot about it from a kind of this organism genetic modification standpoint, but it also can be used as a genetic target detection tool. Again, I'm going to point you to this paper in conservation biology that talks about genome editing generally, but also talks about this as a detection tool in environmental DNA. And then another enrichment approach beyond PCR is capture enrichment. Again, and here we're going to use something like primers, but they're actually called hybridization probes. In this case, in order to draw away our target DNA from our non-target DNA. Here are 2 papers you might go to. This bottom one on the left, I think, is particularly exciting, where they were able to target whole mitochondrial genomes of a fish species and reassemble those bioinformatically. And then finally, I'm going to point out long read technologies. I talked about Illumina sequencing, which is our current dominant approach. But there are some new systems such as the nanopore MinION sequencing technology. These may allow us to get sequence longer amplicons that allow for greater taxonomic resolution, or allow us to do haplotype phasing in order to do some actual population genetics type work from environmental DNA sampling. And some of these systems are also amenable to field application, which might be exciting for a quick turnaround or working in remote areas. All right, so I'm going to take this last slide to summarize the takeaways that I want to communicate from this. I didn't emphasize this at the beginning, but the same rules for controls and clean laboratory practices that David and Matt and Carl talked about in the previous webinars are just as applicable in a multi-taxa system, maybe even more so. Primer selection is critically important and something that you should tailor to your system and your questions. Your database completeness is also really important. This feeds back to influence the effectiveness of your primer selection process and also critically impacts the accuracy of your taxonomic assignment. There are a few rules that exist for bioinformatics, and there are some important inferential caveats that come with multi-taxa approaches, and so it's worth looking at multiple different pipelines and assess consistency across them. And I had a poll too, but yeah, I think I'll put that up and we can do questions simultaneous with that poll, because this poll might be helpful for me as feedback.
And we do have questions coming in.
All right.
So I can ask those. And just a reminder, questions, if you want to ask them, you can type them into the chat using the little bubble icon, or you can raise your hand and then we will unmute you, or we'll call on you. You can unmute yourself and ask them. But first question that we have from Damian Menning: what factors do the primer development programs assess?
Yeah, so each program is going to have some sort of a rule set for imagining what would result in amplification versus not result in amplification. And so if you give it a list of potential templates, it's essentially going to say this is something that will be amplified by these primers, or this is something that will not be amplified by these primers. And it could be a little bit more fine scale than that. For example, the Decipher package is one that I use quite a bit, and it tries to actually say this is what our relative amplification efficiency is going to be for each of these templates. So you're going to have some value between 0% and 100%. So you're actually able to assess not just will this amplify, but how well is it likely to amplify. And some of those software are best for just assessing candidate primers that you already have, but others are going to actually allow you to search for new primers. Give it a list of sequences that you specifically want to target and a list of apps that you specifically want to not amplify, and then it will actually search across those and try and suggest primers that meet your specifications. And I see that Elizabeth Allen provided a link for the Port et al. Paper, so thank you for doing that. That was the paper that showed the decision tree for interpretation after you've done your bioinformatics.
We have a question from Austin Thomas. If you were starting from scratch and building a new bioinformatic pipeline, what software packages would you use or suggest for others?
Well, I think, Austin, my answer would be that I wouldn't start from but I guess one that I have liked recently that I've been working with has been the DADA2 pipeline. So I'm going to put that in the chat box so folks have a record of that, which is an R package. And in part, I like that because it is that happens to be my native object-oriented programming language, so it's convenient. But it also is has a really nice algorithm in there for the ASV construction, which is my preferred approach right now as opposed to an OTU. I'm not saying that ASVs are universally better, but I do like that. And then not starting from scratch, but the maker of that package. I'm afraid I'm forgetting the author's name, but it has some really good tutorials and example scripts. So I think it's pretty easy if you have some familiarity of working with an object program or object-oriented programming language to pick that package up and get a well-working pipeline in place pretty quickly. But I haven't. Yeah, I think that's just one possible starting point. I hesitate to answer that too authoritatively.
Oh, great, thanks. You mentioned earlier that there are some downsides of the ASV approach. Could you elaborate a little bit on those?
Yeah, so I think one potential downside is that you could essentially oversplit relative to the question that you have. So if your question is about are rainbow trout present or not, then you simply need some sequence to represent rainbow trout. You don't specifically interested in multiple, you don't want individual, like multiple haplotypes for rainbow trout. So you might actually have to go through a second compression step where you have ASV1 is rainbow trout, and then ASV5 is also rainbow trout, and ASV7 is also rainbow trout. So I've talked to some folks who will do their ASVs and then they'll actually cluster their ASVs using an OTU approach. Or right now I'm working on a project where we're doing that less, not at that level, but doing taxonomic assignment and then needing to compress after taxonomic assignment. So it's kind of adding an extra step in there.
Cool, thank you. We have another question from Damien. Have you used local BLAST with a custom reference database and high stringency, i.e. 99%?
I think so. I have done a local BLAST with a custom reference database. What I've typically done is actually set the stringency lower for returning a result, and simply return the highest hit. And then after that, you can go through and filter from that. So for example, if you return your percent identity as being any value between, for your top hit is maybe between 90% to 100%, you then could say, well, what would it be hypothetically if I set that threshold at 95? So you remove all of those top hits that are below 95% identity. I guess I would have to maybe chat with Damien more to understand like what your question is related to that. I guess I don't know if I have any general inference about what level of stringency works best, for example, or how reliable that level of stringency is.
Great, thanks. We have time for another question or two if anyone wants to raise their hand or type one in. Yeah, give you a few seconds. All right, yeah, we got one from Elizabeth Allen. Curious if you have had experience with the Minlin and have thoughts on error rates from the Minlin compared to MISEC.
Yeah, I have not personally used the MinION or any of the nanopore sequencing platforms. There is, I think it's still a preprint. Bastian, Egeter, and others recently used the MinION platform for amplicon sequencing, looking for invasive mussels. And I had a chance to talk to them some about their bioinformatic pipeline that they custom built to deal with errors. I can't personally speak. I know that it really seems like the bioinformatics are moving quickly on dealing with errors in the MinION platform. There are also some really interesting systems out there for basically looking at high levels of concordance. So for example, having actually looping together your amplicons. I don't know exactly how to describe that without a visual, but essentially creating a system where you've put a bunch of your amplicons end to end with each other and then sequence them all together. So you're taking advantage of that long read, and then you know that it should be the same amplicon copied over and over. And so you can use consensus among those in order to gain better confidence of your base call. Yeah, I'm sorry that's a little bit of a wishy-washy answer, but I'd be interested in talking to other folks who have direct experience working with the MiSeq and how much they've messed with custom bioinformatics to deal with the higher error rates.
Cool. Great, thanks, Taylor. I think we're pretty good with question discussion here, unless anyone wants one more question. But yeah, let's wrap this up. First of all, I want to thank everyone for attending and for all the great questions. And a huge thank you to Taylor Wilcox. We did record today's webinar, and that video will be posted to PNAMP's YouTube channel in the next couple of days. And it will also be linked on the ETIS landing page. We are going to take a break in December from the Emerging Technology Information Session webinar series, but we'll pick it back up with 4 presentations in January on fish monitoring and assessment, and I'll show that schedule here in a second. And if you haven't already, consider joining the eDNA community of practice, where you can make connections, share information, and start collaboration on work really related to eDNA methods, tools, data handling, and resources. Bonus with the eDNA COP, you do not have to be a DOI employee. And so, yeah, anyone can join. And here's the schedule for that fish monitoring and assessment webinar series, which will be in January. The schedule is also online at pnamp.org and will be published in upcoming PNAMP newsletters. If you don't get the monthly PNAMP newsletter, Amy will put a link in the chat for the sign-up form. And like today, the fish monitoring and assessment webinars will be from 1 to 2:30 Pacific, but they will be held on Thursdays. And that's again in January. And we hope to see you there. And thanks everyone for joining us today. And I hope you have a great rest of the day.